Hi everyone, this is Umair Hassan. I'm a postdoc at the Precision Neurotherapeutics Lab of Dr. Corey Keller at Stanford, and I will be talking about our work on combining TMS and intracranial EEG to investigate the TMS-based neurostimulation. Sorry I couldn't be there today, but I'm hoping to connect with you all in next years. TMS is combined with several measurement modalities to simultaneously monitor the changes it causes in the brain so that we can improve its purpose. For example, it is quite often combined with EMG to measure motor evoked potentials and with EEG to measure TMS evoked EEG potentials or TEPs, or simply just changes in the EEG, and also recently with FNIRS to measure the hemoglobin response, and also with fMRI to measure the bolt activity. All of these different non-invasive modalities to measure the response to the TMS of the brain are extremely helpful and have so much advanced the field, but we also have to acknowledge that there are some limitations. They are very sensitive to the confounds of TMS, for example, the loud click, the somatosensory feedback, and overall the main limitation is a lack of a net spatiotemporal resolution. So we developed this method of applying TMS to concurrently record the intracranial recordings and achieve a net higher spatiotemporal resolution from the brain signals. To provide some background, I worked into this project a bit late. Hiroki Oya from Iowa, he's a neurosurgeon there, did all of the safety testing on phantom brains, and Aaron Boyce, who is a neurologist there at University of Iowa, worked on all of the TMS experiments and human safety basically for the past 7 years now. Corey Keller basically initiated this collaboration between Stanford and Iowa and had Jeff and me doing all of the analyses and now taking up some of the experimental workload as well. So thanks to all of them that we could do this amazing work. So first step was to do some safety testing. In terms of safety, we conducted an extensive testing by addressing three major areas of concern for this combined modality of this TMS-iEEG. The intracranial electrode heating, the second was electrode displacement, and the third one was like the eddy currents that could be generated in the electrodes from time-varying magnetic field. So basically, temperature was measured from a fiber optic sensors at the distance of 10 and 20 millimeters from the TMS coil at a 100% MSO output for a duration of roughly 2 minutes. Luckily, no significant heating was observed from the baseline, and electrode displacement was measured using an HD video monitoring setup. So no electrode displacement was observed at 100% MSO again for that duration. Next, we also try to measure the induced voltages, eddy currents that has actually a linear relationship with the intensity of the stimulation. So we actually fix this intensity to be 100% MSO, and we found out that the voltage of 5 volt at the distance of 5 millimeter from the coil was observed. This is actually 0.3 volts per millimeter per charge density of a phase, and this is well below the units required for the limitation in the safety standards of like, for example, direct intracranial electrical stimulation. So overall, our phantom brain safety tests were very reassuring, and we could move to the human testing. Now at the human side, Iowa has been helpful again, and Aaron has been working on the experiments. We have been implanting a lot more patients recently, and up till this work was published, we had 22 patients. And we did try a variety of stimulation patterns. Actually, I should say Aaron tried a lot of stimulation patterns, intensities, target sites, and durations of stimulation. Overall, no adverse events were reported, of course, beyond what is usually reported after TMS. They had no seizures observed because of the participation in these TMS experiments. Of course, these were epileptology patients, and they had seizures before the day of the or time of the experiment, but not after that, or simply we couldn't trace back it due to the TMS. And then now the first scientific questions we asked were very simple. How does single pulse of TMS apply to the dlPFC affect the brain circuit, basically both locally and also downstreams? So we have our stereo EEG implants, then we also have single pulses of TMS neuronavigated to the dlPFC, to the left dlPFC or right dlPFC both. We applied 50 single pulses at a 0.5 hertz intensity frequency, and compared that to a sham, very simple sham, a flipped coil one. We also then later on added a bit more complicated cutaneous stimulation sham condition into the experiment to have a well-controlled result. And we are measuring the intracranial TMS evoked potentials, or what we call ITEP. This is just one channel from one of the patients, just to show that the waveforms look like and time scale, and we were quantifying the difference between TMS and sham. We saw electrodes that there were no differences, suggesting that this is the sensory component, as is actually TMS criticized for, and then other electrodes that showed no difference whatsoever but zero response. So when you plot this all on an N of 1 brain, I just want to point out that you get is a lot of regions that shows a large TMS effect compared to the sham, so strong ITEPs. And then there are other regions, especially in the bilateral auditory cortex, where we basically would predict where you would see a lot of responses show a similar effect. And of course, we are more interested in the blue regions of here in this graph. So the first question that we asked is, when you apply single pulse TMS, what happens locally, and do you activate circuits or do the neurons respond really to the TMS? And the answer is a little bit. So this figure basically is telling us that there are a lot of electrodes that are colored white, whereas there is no difference between TMS and sham, and there are some electrodes that show the strong effect. So if you zoom in one of these electrodes and see, then you get very nice effects for some and less clear for others, and none for some others. So it's actually only 18% of the electrodes that demonstrated that the effect of TMS are stronger than the sham within, let's say, a specific radius of the TMS coil, so at the local target site. We are actually we were skeptical about it initially, that if it's artifactual or non-artifactual, a combination of both. We are currently diving more deeper here, but the first question we ask is, how does this relate to the electric field distribution? Are there regions that respond those where the electrical field is the highest? So this was a ground map of the electrical field distribution of the IT distribution. So this is again within 3 centimeters of the dlPFC, and the answer is that kind of across subjects, if there was a high E field at that electrode, the electrode is more likely to respond, but the scatter plot was more like 0.4 R square. So it is really only explaining about like the 16% of the effect. It's pretty small, so but it's present. Then next question we wanted to ask was that if we induce any effect into the downstream regions, do we activate downstream regions? And the answer is yes. It's pretty interesting. So both bilateral single pulses TMS to the dlPFC activates anterior cingulate cortex and also the insular cortex more often than dlPFC. That's interesting. When we plot this on a group map, then there are just these regions that light up. So they suggest that single channel responses, at least across these subjects that we are reporting, show that the ACC after a single pulse of the TMS to the dlPFC was activated, and also the insula. So this brought up a lot of questions, including, is this sensory, or is this direct or indirect effect of applying the TMS? Is there some salience network basically causing some sensory motor association here? So we want to tackle that a few different ways by doing some control experiment and analysis. So one of the controls that we did was to compare it with the resting state fMRI correlation. So we were interested in the functionally connected connection between the dlPFC and ACC that we found from ITIP is the same as with fMRI. And the answer is yes. That's been well known that from fMRI that dlPFC and ACC is connected. of course, and that's also true in our patient. But when you put dlPFC seed or you're resting fMRI maps, you get very high positive correlations in those regions that showed the strong ITAPs, specifically in the ACC, but also in the other regions and across the brain. This is true also in the regions that were TMS responsive or had a strong ITAP. So you had a slightly stronger fMRI score. Then we wanted to tackle it in a second different way, which is involving some electrical stimulation, direct electrical stimulation of the brain. Then in these 6 subjects, we applied this e-stim and recorded the cortico-evoked potential, CCEPs, mapped at the electrodes that were in the dlPFC and also outside the dlPFC. And then we asked, do we see a dlPFC to ACC response or dlPFC to insular response in the same way as we see for ITAPs, for example. So this was important because no sensory perception or very little artifact is present in this case of modality. So we would not attribute these to be artifactual or sensory effects. And the answer is that yeah, we do see similar response here. HCC and insula were active. We do get the main stim side effect, of course, at the dlPFC as well. This was again reassuring for us. Then the last control side, our control study that we did was basically just looking at the parietal ITAPs and comparing it to the dlPFC ITAPs. So we did TMS and recorded, we did TMS at both sides, parietal and dlPFC, and recorded the ITAPs. So the question that if this is a pain network that's being activated was basically going to be addressed using this. So the network was basically getting activated indirectly, as we see that the targets also activates the ACC, like the dlPFC target activates the ACC and insula. In this very limited sample size study of n equals to 2, but the parietal stim side did not show it as stronger activation in the ACC and insula, and the TMS and the sham were almost similar, whereas in the dlPFC side, as you see on the left bottom plot, there's differences huge. So again, this showed a little bit of specificity to our method. So I'm hoping that I have shown you that TMS-iEEG is safe and it induces some local intracranial depths within the dlPFC, but also we need to acknowledge that it's pretty low percentage of electrodes that responds to it locally. But this is also somewhat true for other modalities, for example, the bold response, TMS evoked bold response. And it is really not well explained by the E-field distribution. E-field is very high at that local site, but we did reliably evoke downstream effects of the TMS, and they seem to be not sensory. It seems to be related to the direct cortical connections. Of course, as we have to still do a lot more things. We've recently gotten our research grade amplifiers. Previously, these were clinical grade amplifiers. And the current research grade amplifiers have a much more wider range of basically a dynamic range, so which will allow the TMS artifact to not saturate very early. We're also looking at the dose response curve of the single pulse TMS and trying to tease apart the sham. As I said earlier, we're applying like electrical stimulation a bit more into the TMS sham condition. So it's basically auditory stimulation and cutaneous stimulation combined. So together, we are also trying to get the single units recording as the main and original ground truth of the evoked effects that we might measure from TMS. So overall, this is our progress so far. We are diving more deeper here. I would like to actually thank all of the lab members of the Keller lab, Jeffrey, Danny, Ethan, and Chris Klein, and others. And also our collaborators mainly at Iowa. Without their help, this would not have been possible. So Aaron, Hiroki, Nick Trapp, Brand. And I would like to thank my audience right now. And if you have any questions, I might be able to connect with you on my Twitter handle or via email. Thank you very much.
So just before the end, I have to precise something. We were laughing when we realized that in the same session we had two talks talking about ITEPs, but two different ITEPs. So in my talk, I was presenting the immediate transcranial evoked potentials, while those were intracranial transcranial evoked potentials. Well, we should agree a little bit more about how to use acronyms in the future. Thank you again for being here, and thank you for listening to us. Thank you.